All right, so we're still in chapter 16 and we discussed the sympathetic division in the first video. And now we're going to go into a little bit more detail about the parasympathetic division. Now the parasympathetic division is also called craniosacral because it is located up in the brainstem and at the bottom of the sacrum. Autonomic nuclei are in all parts of the brainstem and in the lateral horns of sacral 2 to sacral 4 regions. And we're going to have our ganglionic neurons. Now, they're not in a chain close to the spinal cord like we saw with the sympathetic division, but rather they are all the way out at each individual organ, either beside the organ or actually sometimes inside the organ that is going to be controlling it. So that means that the first neuron, the preganglionic fiber, leaves the lateral horn of the spinal cord, joins the spinal nerve, and has to go all the way out to the organ before it finds the ganglion to synapse with the postganglionic neuron. That means for the parasympathetic division, we have long preganglionic fibers and short postganglionic fibers. So here's our picture. Notice how different this one looks from the autonomic system diagram. So we have all of these nerves coming off of the brainstem. Okay, notice that once we get into the actual spinal cord, we have no nerves coming until sacral region 2. And each one is an individual pathway. Once in a while, you can kind of control 2, but for the most part, each of these is an individual pathway with the exception of this one. We're going to talk about this one in a second. That gets us out to each individual organ. Now, if we're under parasympathetic control, we're not in an emergency. We are in rest and digest. So I don't have to send an action potential out by emergency broadcast system to all my organs. Instead, I can fine-tune the control of my lacrimal gland or the pupil of the eye or the salivary glands. So I don't have to have a giant bunch of connections between these parasympathetic nerves. Now, with the exception of the vagus nerve. Now, the vagus nerve is one of our cranial nerves. You remember it's nerve 10. And it is the parasympathetic controller of most of the organ systems. People laugh because I actually have a favorite cranial nerve. I know that's weird. But the vagus nerve is amazing. See, when I was little, I've always been fascinated by the body. And I was not always, the most polite. Whenever I saw someone in a wheelchair, like a quadriplegic, I really wanted to ask them, how are you alive? I know it would be so rude to say that, but in my brain, in my child brain, I knew that if they couldn't move anything below their neck, I wanted to know, how is their heart still beating? How do they digest food? How are they able to, urinate or defecate if they don't have control of any of those body functions? And I learned, as I studied anatomy, that is through this cranial nerve. This vagus nerve 10 is 75% of the parasympathetic output, and it never enters the spinal cord. It comes off of the medulla. So if your spinal cord is severed, you still have a connection between your brain through the vagus nerve to all of your major organs. So you can still keep your heart beating and your lungs breathing and your digestive system working. You can even control the smooth muscles of the urinary bladder. And believe it or not, people who are quadriplegics can still have children. Okay, they can still, a woman can be, if she can get pregnant, she can still deliver her baby. Isn't that crazy? All because of this special vagus nerve right here. Okay, so let's go on. So we have the ganglia, and they're either terminal or intramural. Terminal means that the ganglion is outside of the organ, but very, very close by. So it's near the target organ, and these are usually in pairs. Intramural means actually in the wall. And so an intramural ganglion is inside the organ that it is controlling. It's a bunch of interconnected masses and clusters of ganglion cells. Again, the vagus nerve, the one I was just explaining, provides 75% of all parasympathetic outflow. And so it controls things in the neck, in the thoracic cavity, and in the abdominopelvic cavity, including the large intestine. And the branches of the vagus nerve will intermingle with fibers of the sympathetic division. Again, there's our map. Okay? So if you stimulate the parasympathetic division, you're going to see opposite effects of the sympathetic division. Instead of dilating pupils, you're going to see constriction of pupils, and you're able to focus on near objects. Your digestive system is going to be ramped up. So we're going to have more secretion by digestive glands. We're going to have more absorption of nutrients from the digestive system. We're going to have the early stages of sexual arousal controlled by the parasympathetic division. So here's a little hint. If you are not able to be in a relaxed state, you cannot be sexually aroused. You have to trust the person you are with in order to be under parasympathetic control. We're going to have increased smooth muscle activity in the digestive tract. We're going to be able to stimulate urination and defecation. We're going to have constriction of respiratory pathways and reduced heart rate and reduced force of heart contraction. So think about all the things when you're just kind of relaxed and chilling out, the things you would expect your body to be doing. Now, unlike the sympathetic nervous system where we had cholinergic and adrenergic neurons, all of the neurons within the parasympathetic division only use acetylcholine. So they are all cholinergic neurons. Now, that doesn't mean that all cells respond the same way. In fact, cells can respond differently depending on which receptors they have or which second messenger system they use. So the effects of parasympathetic stimulation of cholinergic receptors are localized and short-lived because we usually get rid of our acetylcholine, that's our neurotransmitter, by using that magic enzyme acetylcholinesterase. Remember, ASE means enzyme, but the cool thing here is we have erase in the name. It tells you what it erases, acetylcholine. And any acetylcholine that diffuses into surrounding tissues is also inactivated by tissue cholinesterase. So sympathetic division, remember our fight or flight, not parasympathetic, has widespread effects. It's emergency broadcast all or nothing. The whole system responds. Parasympathetic, I can pick and choose and send individual signals to different organs through different pathways. The sympathetic division has two sets of sympathetic chain ganglia on the right and left of the vertebral column, 3 collateral ganglia in the abdomen, and 2 adrenal medulla. And again, I keep drumming this because it's going to come up on tests a lot. Sympathetic is short preganglionic, long postganglionic. Parasympathetic is long preganglionic, short postganglionic. In the sympathetic division, we have extensive divergence or spreading of a signal. Preganglionic neurons release acetylcholine. Most of your postganglionic ones release norepinephrine, noradrenaline. And the effector response depends on the second messengers. So here is a really good graphic. There's a lot of information in it comparing the sympathetic to the parasympathetic divisions, talking about the lengths of the neuron fibers, talking about what kinds of ganglia they are, which neurotransmitters they secrete, and what their targets are. So this will be a really good thing to study and understand these two divisions. Now, like I mentioned in the first video, most of your organs have what's called dual innervation, which means they can receive input from both the sympathetic and parasympathetic division, but they have to take turns because they often do opposite things. So if the parasympathetic division is in charge, then the sympathetic cannot be in charge. They have to take turns. Sympathetic innervation reaches the same structures from the superior cervical ganglia sympathetic chain that the parasympathetic ones do, and they travel by cranial nerves. So, the anatomy of this dual innervation. Autonomic plexuses. Remember, a plexus is an intertangling of neuron pathways. So we have networks in both the thoracic and abdominal pelvic cavities, and we're going to mingle sympathetic postganglionic fibers and parasympathetic preganglionic fibers. And they're also going to travel with blood and lymph vessels, and they're going to go to the organs. Okay? Now, autonomic tone. You have smooth muscle all through, controlled by the autonomic system, right? So you're going to have some stimulation of the motor neurons, even without active stimulation from the nervous system. So you have a resting level of activity, and this is important. The nerves maintain your background level, so they are always at the ready. They don't have to come on. We can just fine tune whether we need to tone them down or push them up. Okay, so it's really important where you have dual innervation that you have them both have a background amount of activity. It's even more important where you only have one of them. For example, blood vessel dilation and constriction is controlled solely by the sympathetic division. So you want your blood vessels to have a little bit of constriction going on so that they have the ability to either constrict more or to dilate. If they were at one end of the scale and had to get to the other end of the scale quickly, it would be much harder. So you kind of keep them, if it was a 1 to 10 on constriction scale, you kind of keep them between 3 and 4. So they can go backwards to dilate or they can go forwards to constrict more. The heart gets dual innervation, so controlled by both parasympathetic and sympathetic. Anytime the parasympathetic stimulates the heart, it releases acetylcholine and it will slow the heart rate. If adrenaline or noradrenaline is released by the sympathetic division, it accelerates the heart rate. And the weird thing is, actually, both of them are released all the time. So whoever is secreting more wins. Parasympathetic division dominates at rest, and any crisis speeds up the heart rate by stimulating the sympathetic secretion of adrenaline, which overrides the acetylcholine. Some organs only have one division. For example, I already told you, sympathetic control of blood vessel diameter. All right, so that's the end of the parasympathetic and sympathetic part of the chapter. I'm going to very quickly finish off this chapter with the higher order brain functions. There's not very many slides here. This is functions of the brain that are the sum of its parts. Like it takes many brain regions to accomplish these particular jobs. So they require the entire cerebral cortex. It can involve both conscious and unconscious information processing, and you get to adjust this over time, which means you're not born with these higher order functions, you develop them. So they are not innate fixed behaviors or instincts. Instead, they are learned behaviors. So things like memory. Memory is technically defined as stored information that's gathered through experience. Now there's two categories of memory. There is fact memory and there is skill memory. This class is going to be mostly fact memory, which sucks because fact memories are the hardest to recall. Skill memories are motor behaviors, things like riding a bike, walking and running and all those things that your body learns to do and learns the patterns of and can do without having to think about it. So you incorporate it at an unconscious level after you master the skill. And most of skill memories are stored in the brainstem and in the cerebellum. Fact memories are stored in the cortex of the cerebrum. Complex skill memory, so it's a very hard thing to do, can involve integration of the basal nuclei, the cerebral cortex, and the cerebellum. Now, fact memory can be separated into short-term and long-term memory. Short-term memories are things that have happened right now. They can be recalled immediately. They will not stay in short memory long. They are little bits of information. There's actually some fact that we can't really hold more than 9 pieces of information at a time. And in order to keep the information that has been brought into short-term memory, you must perform some function that moves it from short-term memory to long-term memory. And that movement is called memory consolidation. Now, there are 3 things that will move something from short-term memory over to long-term memory. There is repetition. So if you repeat it over and over, you expose yourself to it in different contexts. So for example, you read the information in your textbook, you see it printed on the PowerPoint slide, and you hear it in my voice. That's repeating it 3 times. That's going to help you move it. Okay, so repetition. Relation. Relation is something that I really try to do in this class. If I have a new bit of information, I try to give you an example and link it to something you already know. For example, when I said, when you hear the word autonomic, I want you to think automatic. So I'm relating those 2 terms. And so now this term you may not have been familiar with, autonomic, is related to something that you do understand. So repetition, relation, and then I kind of cheated on the third R, really strong emotions. Really strong emotions will cause you to move something from short-term memory to long-term memory very easily. For example, think about your best day you've ever had. Could have been your graduation day, your wedding day, the day your child was born, okay? Those strong emotions, that a lot of details of that day are imprinted in your long-term memory. Also, now think about your worst day. Same thing applies, right? A lot of details moved over to long-term memory because of the strong negative emotions that you associate with that event. So really strong emotion, repetition, and relation will all help you consolidate memories and move them over from short-term to long-term. Now, within long-term, we have two types. We have secondary and tertiary. Now, tertiary, I'm going to start there. Tertiary memories are permanent. Again, these are often tied to very strong emotional events. Your best day, your worst day, memories of your mother, your pet, all of those things are going to be tertiary memories, and they don't go anywhere. Secondary memories are ones that you use for a while. You had to take it from short term to long term, but if you don't continue to use them, eventually they will fade. Now, if you have seen the Disney movie Inside Out, you've kind of seen a visual representation of secondary memories. Her little imaginary friend that she used to use her brain to imagine and think about all the time, as the little girl got older, the memory of him started to physically fade in the cartoon. But that's what happens to secondary memories in long-term memory. If they're not recalled, they eventually go away. Okay, so again, we're talking about mostly the cortex, that thin layer of gray matter on the surface of the brain. And so everything that you see and hear and experience is coming in through your senses, and it sits first in short-term memory. If you repeat it, it tends to promote retention in short-term memory and helps consolidate it. If you do not consolidate anything that comes into short-term memory, it is permanently lost due to neural fatigue, shock, or interference by other stimuli that take its place. If you move it over through consolidation to long-term memory, it first starts out as a secondary memory. Okay, fact memory stored in the cerebral cortex, skill memory stored in the cortex and cerebellar cortex. And then if you use it a lot or it's very tied to very strong emotion, it can become a tertiary memory. If it is not moved from secondary to tertiary, it can also be temporarily or permanently lost. Okay. The second higher order function is called consciousness. Not conscience, not good or bad, but conscious, like how awake are you? And this is a huge scale. Everything from alive but completely unconscious, that's called being in a coma, to having experienced an emergency where you are the most alert, the most awake. So this is your degree of wakefulness. And it's related to how many electrical signals are traveling through your brain at any one time. If we reduce the number of electrical signals and depress it, okay, then we're going to reduce your consciousness. Deep sleep is just above a coma, and it's called slow wave or non-REM sleep. Your entire body is relaxed. You actually have minimal cortical activity in your brain. Your body reduces its metabolic rate by 30%, and you're supposed to be temporarily paralyzed. We'll talk about that when we get to REM. Okay? Rapid eye movement sleep is where your dreams occur. Now, if you did not temporarily paralyze your body during REM sleep, your body would carry out all of the things that you are dreaming about. In fact, anybody who doesn't completely paralyze themselves minus their eyes and breathing apparatus will perform sleepwalking, right? So REM sleep happens where it's where active dreaming occurs. It causes changes in blood pressure and your respiratory rate. But if you were to look at the brain of someone who is in REM, it looks very much like an alert, awake person. You are less receptive. It's harder to wake you up. Your muscle tone is decreased. You have intense inhibition of voluntary motor neurons, and your eyes move rapidly. So in sleep, you alternate between REM and non-REM. Now, REM periods are much shorter, and you always start with REM sleep. So if you've ever felt that like falling off a cliff feeling as soon as you close your eyes, it's because you immediately started to dream. And you go from REM, and then you go to non-REM, and there are 4 stages of non-REM that go deeper and deeper and deeper, and then you got to work your way backwards from the 4th stage of non-REM, 3rd stage, 2nd stage, 1st stage, and then back to REM. So REM starts out at only 5 minutes the first time, and then it will increase and increase in length until, if you're getting a full 8 hours sleep, your final REM period is about 20 minutes long. During this time, your brain is doing a reboot. It is erasing your short-term memory, and it's making you have a clean slate for the next day. You're going to have minor changes in physiological activities of organs, but major protein synthesis in neurons, which represents storing important information and getting rid of non-important information. So if you go for extended periods without sleep, then you actually decrease your mental function. So don't stay up all night the night before a test. It actually decreases your ability to recall information. So here are EEGs, or electroencephalograms, of an awake person on the top, somebody in REM, and looks very much the same, and then somebody who is in non-REM, deep wave sleep. So you can see down here is the restorative sleep. It's the brain reboot sleep. And here's a typical 8-hour sleep cycle. So you go from awake to dreaming to transition to deep, and then back up. And you go back up several times. So you should dream 1, 2, 3, 4, 5, about 6 times within 8 hours. Okay? And as you go on, you get your deepest sleep early on, but then after that, we don't get quite down into deep sleep. And unfortunately, as you get older, you go into the deep sleep time even less. Isn't that depressing? I think something like after age 60, you almost never get into the deep sleep stages. Okay, so how do you get awakened from sleep or arousal? It involves the reticular formation, which is part of the reticular activating system. And it sends out electrical pulses. It also connects with sensory, motor, and integrative nuclei. Okay, so you're going to send out these little electrical pulses through the reticular activating system. And the more frequent these pulses, the more alert and awake you are. The less frequent, the more towards the unconscious state you are. Sleep can be ended by any stimulus that activates the reticular formation and the reticular activating system. Now, one single startling doesn't usually awake you. It usually takes multiples to get you awake. Effects of a single stimulation last less than a minute, and if you go back to sleep, it's because you don't get any additional interruption. If you stay awake, it's because you have positive feedback from the cerebral cortex or the basal nuclei. Now, you use several neurotransmitters to control your sleep-wake cycles. Okay, you're going to have some that promote deep sleep. Serotonin is one. To get us into sleep, we use melatonin coming from the pineal gland. We'll learn about that in the endocrine chapter. And then serotonin keeps you asleep. Another group, though, is trying to maintain your awake, alert state, and it uses norepinephrine. So if you're very fearful and you're in fight or flight, this norepinephrine can override the serotonin and keep you awake. So here's a diagram of the reticular activating system sending out pulses. And that's the end of the chapter. All right, guys. So that is chapter 16. If you have any questions, feel free to contact me through text or email. If you text on the cell phone, please go ahead and tell me who you are so I know how to respond. All right, guys. Happy studying.